Okay, well, thank you for the invitation to come and speak. I'm excited to come back to the source, as it were, from some of the algorithms that we have been using in my lab to try to understand neural circuits. My talk's going to emphasize the problem that we're trying to tackle, which is one of the major problems in neurobiology, and that's how sensory systems decode sensory information. What I mean by decoding is that there are objects out in the external world that produce sensory signals, and one of the jobs of the brain is to reliably detect what those distal objects are. And we are trying to tackle this problem in the realm of speech recognition. It's a particularly relevant problem for a number of reasons. And to illustrate what I mean by this decoding problem, I'm going to show you a spectrogram of somebody saying this English phrase, "any bright young man who still thinks." So spectrogram is a time-frequency representation, time on the x-axis, frequency on the y-axis, power indicated by the darker colors. And you can notice some things from this spectrographic representation, which the first thing is that the segmentation is not what you hear. You hear gaps between the words that I'm producing. In the spectrogram, words don't actually have gaps between them. Our auditory system is automatically segmenting this sound stream, this continuous sound stream, into a sequence of discrete objects that we call phonemes and syllables. The second thing to note is that the same phoneme, so phonemes are acoustic objects that give words meaning. The same phoneme I in still and in thinks actually has a very different acoustic representation. So in still, this upper formant is decreasing. In thinks, the upper formant is increasing. And this demonstrates a phenomenon known as co-articulation. When I'm speaking rapidly, the articulators are in my mouth and in my vocal tract are reconfiguring themselves in anticipation of the sounds that I'm going to be producing. And that actually influences the spectrographic quality of the sound that I'm producing right now. So still is moving down because I'm anticipating that sound that I'm going to make later. There's other reasons why the same distal object, the same phoneme, can produce many different acoustic representations. So there's variations in pitch and duration, variations between speakers, and then variations in different environments. So when you're speaking in a noisy environment, the spectrogram is now covered up with lots and lots of spectrograms from people all speaking at the same time. But the auditory system has solved this problem of determining what the distal object is that's giving rise to those different sensory representations. The flip side of the sensory decoding problem is that different distal objects can give rise to very similar acoustic percepts. So this is illustrated by this spectrogram here, "Did you hit it to Tom?" And then down here where we've substituted some of the syllables with nonsense syllables, so Dom instead of Tom. You'll notice that these two very distinct acoustic objects, Tom and Dom, actually have very similar spectrographic properties, especially in the later part right here. What differs is actually the timing between the onset of the consonant and the initiation of the vowel. That can differ on the order of about 10 milliseconds. Lieberman and Mattingly did a number of experiments back in the 50s using artificial speech synthesis to explore this. So they used this device where they could actually paint spectrograms on a slip of cellophane. And so they could systematically vary acoustic features of the speech signal and then ask people, "What do you hear?" So this is a sequence of sounds that ranges from boo to goo. And the only thing that's changing is the shape of this formant transition. And even though this is a continuous transformation in the proximal domain, in the perceptual domain, people hear very sharp transitions between b, d, and g as you move between different sections of this continuum. So different distal objects can give rise to similar proximal representations. So this is sort of an inverse problem, and it's an inverse problem that the brain has solved very effectively. There's been a lot of interest in the technological realm in trying to mimic this ability of brains to parse speech. And there's some degree of success and some degree of failure. So sometimes Siri misunderstands the questions or the statements that you're trying to give. And so there's a lot that we need to learn about how auditory systems solve this decoding problem. So the outline for my talk is I'm going to talk about why we use songbirds as a model for this process. I'm going to talk about an observation that we've made, which is that one of the features of the auditory system seems to be the use of very diverse neuronal dynamics. And then I'll talk about how we're using data assimilation to try to understand the diversity of neural response properties and how they contribute to sensory processing. So Henry has given a good introduction of the birdsong system. I'm going to point out a few additional features. So one of the reasons why we use birds is that they produce these complex signals called song that are essentially their form of speech. It's a social signal that they use to advertise themselves. And so one of the things is that male birds will try to impress female birds by singing, but different individuals have different songs that are unique, and they identify themselves by singing. So birds are faced with the same decoding problem of trying to identify the individual that's producing the song from the acoustics of the song. Here's a spectrogram of a zebrafinch song. I hope that this plays. So it doesn't quite sound like human speech, but birds seem to like it. The other reason that we study songbirds is that the ontogeny of the development of song production is very similar to the ontogeny of human speech production. So there's an early phase, so singing requires a model. Birds have to hear another bird sing in order to form the appropriate template of the song that they're going to produce. That takes place during an early sensory learning phase that corresponds in humans to the first year of life. The language that you speak and your perceptual abilities are shaped by your early sensory experiences. And there's also a sensory motor phase where the bird tries to match its motor production to the sensory template that it's formed. So the formation in both humans and birds of this sensory template is critical to later production and perception. So there's also a clinical reason why we want to understand this process of speech decoding, because if this doesn't happen properly, it can have effects on both language production and language perception. So the other reason that we study birds is that there's a lot of similarity in the processing pathway, in the neural processing pathway that processes song to the pathway in humans that processes speech. So as in all other vertebrates, sound enters the ear. It's transduced into neural signals in the cochlea. And there's a pathway that goes from the cochlea and ascends from the periphery into more central areas. So you can trace this information as it flows between these different nuclei. And as the neural information is passed through each phase of this processing, the brain is extracting new pieces of information. It's performing this decoding process. What I mean by hierarchically is that each stage, the representations are transformed into more abstract forms. So I'm actually going to illustrate this in the visual realm because we have a slightly better idea of what's going on with visual information than we do with auditory information. The idea is that as you move from the periphery, in this case the retina, to more central regions, cells become responsive to more global, more abstract features of the sensory stimulus. So the retina is going to have little patches that respond to light and dark. But as you ascend the visual processing pathway, cells become selective for oriented bars and for contours. And the idea is that our ability to decode that this visual signal means A and this visual signal also means A is that they have the same configuration of contours. Distinguish it from symbols like B. So this model implies a sort of feed-forward process where information is transformed at each stage. And one idea about how that works is that there's a lot of convergence in the neural architecture as you move forward from each stage. So here I've sort of represented, some sensory continuum, and each neuron at a successive stage is receiving input that samples from a range of different areas on this continuum. So the idea is you can get composite receptive fields that add together the information that's coming in from lower stations. But we also know that these neurons are part of diverse recurrent networks that are all very strongly interconnected. So this neuron is part of an ensemble of interconnected neurons that are all mutually exciting each other. And we also know that there are different types of inhibitory neurons that actually suppress activity. And this can lead to some interesting dynamics, some winner-take-all dynamics, such that the green ensemble here can suppress the activity of the blue and the red ensemble to produce a percept of a particular stimulus. So we know a fair amount about the structure of the auditory system, and there are some very informal models about how information is transformed by it. But the formal models are actually lagging the informal models. And so I'm going to show you the kind of dominant model in the field of neural processing, which is the idea that neurons are basically doing linear summation. They're taking a bunch of different inputs, synaptic inputs, they're weighting them, summing them together, comparing them to some threshold, and then producing an output. So this is very similar to that feed-forward model that I showed you of the circuit. Doesn't take into account recurrent connections. And when you translate this model into thinking about how neurons respond to sensory information, we have this model called the linear-nonlinear Poisson model. The idea is that some sensory stimulus, say a spectrogram, is convolved with a receptive field that has different channels for different frequencies. This produces a signal that then is passed through a static nonlinearity. And then to take account for the fact that neurons respond differently on different trials, that there's some randomness in the response, a Poisson process is used to transform the estimated rate into an actual spike train. So this is a very simple model, and I'm going to argue that it's an overly simplistic model. One of the things that is critically missing from this is that there are no dynamics. We know that neurons are dynamical systems, and yet our best model for trying to represent what neurons are doing computationally doesn't have any dynamics in it. It also doesn't have any sort of circuit connections that represent these recurrent networks. So I'm going to show you some evidence that this model is wrong. I'm going to show you why it matters that it's wrong, and then I'm going to talk about some of the ways that we're using data assimilation to investigate more complex models of this aspect of processing. So a brief word on the techniques that we use. We use slice electrophysiology. We take the brain out of the bird. We cut a slice that passes through this auditory region called CM. We put the slice under a microscope and stick a very fine-tipped glass electrode in there, and we can patch onto a cell. This is a high-powered magnification of this process. So the tip of the electrode is about a micron across. It's hollow. You can bring it up to the cell, and it will form a molecular bond with the surface of the cell, and then you can suck out the little bit of membrane and obtain electrical access to the inside of the cell. So now you can measure the voltage difference between the inside of the cell and the outside, and you can pump currents in and out. You can drive the system. And the thing that people usually do when they want to understand what these cells are doing is they inject some current in a step. So this is data from a very old paper from McCormick et al. And here's a current step, and here's the response of the neuron. So most neurons rest in a fixed point state. They have a fixed resting potential. As you increase the current, they go through a bifurcation. It's usually a Hopf bifurcation. And then they enter a limit cycle, which is the emission of these spikes. And as you change the amplitude of the current, the rate of the frequency of the limit cycle increases. So this is the kind of standard model of how neurons behave, or the standard observation of how neurons behave, and it actually relates back to that linear-nonlinear model because this is a fairly linear process. As the amplitude of the current increases, the frequency increases in a relatively linear fashion. But we actually find when we patch onto neurons in zebra finches that these dynamics don't apply. So here is an experiment done by my graduate student Andrew Chen. Very similar. He injected a step current, but rather than the neuron entering a limit cycle, it actually fires a single action potential and then stops firing. And when we record from a variety of neurons in this area, we find that there's actually a large diversity of dynamics. Some cells are highly phasic, some cells are more tonic, some cells are intermediate. And if you look across the entire population, what you find is a pretty broad diversity. Some cells with very tonic firing patterns and some cells with very phasic firing patterns. So this model of cells all being the same and all having regular spiking dynamics is not correct. So we wanted to ask, do these dynamics matter? What effect do they have on sensory processing? So then instead of injecting step currents, we injected a very complex current waveform, recorded the spiking responses of the neuron. These are two different neurons. This is a tonic neuron and this is a phasic neuron. These are raster plots that show the spike times emitted by those neurons. And what we could do with these data is calculate the coherence between the input and the output, between the current and the spiking output. And we find that the phasic neurons, this green curve right here, have a peak in the coherence spectrum that's at around 20 hertz. And they have fairly strong low-pass attenuation. In contrast, the tonic neurons have their peak coherence at a much lower frequency, and they have much lower low-pass attenuation. And there's a direct linear relationship between the duration of the cell's firing and the peak frequency and low-pass attenuation. So coherence is related to mutual information, and so what this result says is that cells transmit information about events that are happening at different timescales. We think that's the function of the diversity, is that song and speech has many different timescales, and neurons are tuned to different aspects of that. So to try to formalize that intuition a little bit more, we have worked on developing, on taking this linear-nonlinear model and adding dynamics in. So replacing this random history-independent Poisson process with a dynamical model of the system, similar to the one that Alon just showed you. So membrane voltage, the change in membrane voltage as a function of time depends on these currents. But we're saying that one of these currents, the stimulus current, the driving current, is the convolution of the stimulus with the receptive field. So we now have a synaptic receptive field that represents what the neuron is tuned to, and then some dynamics that transform that into a spiking output. We are not using the same model that Alon described, in part because we're still at kind of the proof of principle stage for this. So we're using a much more simplified, reduced model called the MAP model, which is actually from the Shinomoto group at University of Kyoto. And instead of having this collection of currents that all contribute to the cell's dynamics, what this model says is that there's these multiple adaptation, there's a threshold. The input current passes through a leaky integrator, and then when it crosses a threshold, then the cell emits a spike. And there are a small number of parameters, these alphas and taus, that essentially control the behavior of the threshold. And you can get a variety, the important thing about this model is you can get a variety of different types of dynamics from it without having to dive into a lot of biophysical detail. So you can get regular spiking, fast spiking. This figure shows a whole family of different types of response properties of dynamics that you can get. So importantly, you can get phasic spiking and you can get tonic spiking from this model simply by moving between different regions of this parameter field. So now we have a model that is a sensory processing model but includes neural dynamics. And so we can ask, do those dynamics matter? This is a simulation experiment that was done by tech in my lab, Tyler Robbins. These are two different neurons, two different MAP models. One is phasic and one is tonic. They have the same synaptic receptive field, and they've been stimulated with the same stimulus, so the same song. Here's the response of the phasic neuron. Here's the response of the tonic neuron. You can see by comparing the average response rate of these two different neurons that there are some significant differences between them. So the dynamics do affect the output of the cell. And they affect it, it's rather subtle when you just look at this, at these raster plots, but if you take the spike times and you do a standard linear regression to try to recover what the cell's receptive field is, what it's responding to, you see that the dynamics actually have a huge effect on the response properties of the cell. So here's the input receptive field, here's the output receptive field of the phasic cell. And you notice that it's become much, much more complex. It's now responding to 2 different frequency bands, and it's also quite different from the tonic cell. So those 2 cells, by this very small difference in dynamics, have very distinct sensory response properties. So emboldened by the success of this model, we have now turned to see if we can use it to characterize neural diversity in living animals. So instead of just doing simulations and slice recordings, we would now like to use a technology where these large arrays of silicon probes with large numbers of recording sites can be inserted into a bird brain. Bird can be behaving or can be quietly listening to a stimulus. You can record neural activity on a wide range of channels. You can detect through extracellular means spikes, and then convert those spikes into a raster of many different neurons responding at the same time to the stimulus. So now we have information about not just a single neuron, but a whole circuit of neurons, and we would like to understand whether these differences in dynamics actually translate into whether we can detect them in the spiking behavior of the cell. So we did a couple of simulation experiments. This is a twin experiment using this map model. We simulate the response to a complex current, like the ones that Alon showed you, calculated the spike times of the neural response, and then I'm not going to go into the details of how we did the DA, but we use a Markov chain Monte Carlo approach, so we actually get a posterior distribution of the parameters. And you can see that we can very tightly constrain the parameter estimates of the map model just using the spike times alone. And this maps onto a specific region in the parameter space, which is where we can recover the parameters that we started out with. So we can then turn back to this D-Sturf model and try to recover the parameters that went into the model. And you can see from these little crosshairs that the posterior distributions are lining up on the correct locations. We can recover the synaptic receptive field. So you can see that the D-Sturf model does a much better job of recovering the input receptive field than this standard linear regression model. These are the correlation coefficients for the prediction. And we can use this with real recordings. We have a data set, a publicly available data set of extracellular recordings from zebrafinch brains. So we can they consist of spiking responses to a variety of different stimuli. We can estimate the dynamical models, the receptive fields. And so we've applied this method to a large collection of these recordings, and the data are still fairly preliminary, but what I want to point out is that these 2 parameters, beta and omega, have a bimodal distribution. So there's a group of cells with a low value of omega and a group of cells with a high value. There's actually a correlation between beta and omega. And if you map that back into this parameter space, you find that this smaller group of cells is actually, those are phasic neurons. So the take home is that using spike times alone, using just information about when the cell is firing, we can actually recover information about the intrinsic dynamics of the cell. We can assimilate that data into a model of the cell that contains those dynamics. So this offers us a path for using data assimilation as a method of characterizing neural dynamics. We can do it in this very phenomenological way. And what we would now like to do is to extend the work in a couple of different areas. The first is to start looking at circuit models. When we have these large scale arrays, we can record from hundreds of neurons at once. Now we can start to infer something about the connectivity of the neural network and start thinking about the dynamics of the network that are doing this computation. And importantly, if you know anything about this technique of optogenetic manipulation, we can actually understand in a quantitative way the effects of this new tool that allows direct drive of many different neurons in a circuit. And then we'd also like to turn back to more biophysical models like the ones that I worked with Henry and Alon on, And the important thing is, as Henry pointed out, is that when you have a biophysical model, those channels are real proteins that have real pharmacological properties. And so this is a path for understanding what the effects of drug treatments are and the effects of disease on circuit dynamics and on and how those proteins affect the way that circuits do their computations. Let me briefly thank the people who did the work. So a lot of these techniques came out of my collaborations with Dan Margoliouth and Henry Barbanel and Alain. And then Andrew Chen and Tyler Robbins are the people who did the recordings and the data analysis, respectively. And again, thanks for the invitation, and happy to take questions.